Good afternoon to all the BIPC students. So, here we are with short answer type of questions, last session, where we are going to cover up the questions today from the chapters biological classification and cell cycle and cell division. So, here if you see, first question is about schematic representation of primary, secondary and tertiary structures of hypothetical polymer using protein as an example. So, with regards to your exam also I am telling you, see, in your exam if you observe both EAP set and NEET, all the levels of questions are important. Primary structure, secondary structure, tertiary structure, very important. So, here if you see, first primary, secondary, tertiary. Here this primary structure is what? Linear sequence of amino acid. This will give positional information of amino acids. Positional information of amino acids. Secondary structure, this is local folding of chain into what? Alpha helix and beta pleated sheets. And tertiary structure will give you overall 3D folding of a single polypeptide chain. Next, primary structure is stabilized by only peptide bonds. Secondary structure has both alpha helix and beta pleated sheets. Alpha helix and beta pleated sheets. These are stabilized by hydrogen bonds between backbone atoms. Next, tertiary structure is stabilized by hydrogen bonds, ionic bonds, disulfide bridges and hydrophobic interactions. Fine, right? Quaternary structure will also be there, for example hemoglobin, but here it is not included, that is fine. Now, primary structure will determine higher level structures, secondary structure will give initial 3D framework, tertiary structures, this will determine proteins function and activity. Here if you see examples, this will give sequence of amino acid in insulin. Secondary structure, example is keratin, alpha helix and silk fibrin beta sheet. Tertiary structure, example is myoglobin. Clear till here? Any doubt anywhere, ma? Now, this you can see, this is the primary structure, followed by secondary structure, that is alpha helix, and tertiary structure which is given over here. Next, nucleic acid will exhibit secondary structure described through Watson-Crick model. Okay, describe this through Watson-Crick model. This has appeared in multiple years like 2021, 2018, March 2015. And this is important for NEET and MCET as well, that is double helical model which is given by Watson and Crick, which explains the stable form of DNA, that is bDNA. Clear, right? So, here DNA will consist of two long polynucleotide chains which are twisted around each other. How they are twisted? In right-handed double helix. These are twisted in right-handed double helix. The two strands will run opposite directions. The two strands will run in the opposite directions. One from 5 prime to 3 prime and the other from 3 prime to 5 prime. Fine, right? One is from 5 prime to 3 prime and the other is from 3 prime to 5 prime direction. Next, each strand has backbone which is made up of alternating deoxyribose sugar, which is a pentose sugar, and phosphate group. Fine, right? So, here if you see, the backbone of this double helical structure consists of sugar and phosphate group, where nitrogen bases are at the center. These nitrogen bases are binded by what? Hydrogen bonds. Next, these bases if you see, adenine, guanine, thymine and cytosine, they project inward perpendicular to the backbone. Okay, they will project inward. See, these are sugar phosphate bonds which will form the backbone of the double helical model, where at the inner side if you see, nitrogen bases will be present which form base pairs. They will project innerwards. Adenine pairs with thymine by how many hydrogen bonds? 2 hydrogen bonds, and guanine pairs with cytosine by 3 hydrogen bonds. So, to cleave guanine and cytosine bond, it requires more energy than to cleave the bonds between adenine and thymine. This complementary base pairing will ensure accurate replication. because adenine always pairs with thymine, guanine will always pair with cytosine. Always pairs with thymine, guanine always pairs with cytosine. The helix will maintain a uniform diameter, the uniform diameter of 20 angstroms. One complete turn of helix will have just 10 base pairs with total length or pitch of one complete pitch or length will have how much angstroms? What is the length? 34 angstroms. The distance between two successive base pairs is distance will be 3.4 angstroms. Fine, right? Now, each base pair is rotated 36 degrees, relative to next along the helix axis. The helix shows two alternating grooves, one is called as major groove and second is minor groove. Major groove is deep in the B type DNA, minor groove is shallow in B type DNA, which serves as binding sites for proteins and enzymes. The double helix is stabilized by hydrogen bonds between complementary bases and base stacking interactions. This structure is described by what? Watson and Crick is the most common form of DNA in the cells, and these are known as bDNA. Clear till here? So, first I will always recommend you to draw the diagram double helical structure, then start elaborating the double helical model of DNA. Also mention about complementarity and anti-parallel nature of the DNA. Till here it is clear, right ma? DNA secondary structure, Watson and Crick model. Till here everything is fine, right? Now, write a brief account on polysaccharides. Okay, polysaccharides, first you have to know about the types of, you have to mention about the types of polysaccharides. We have monosaccharides, disaccharides, oligosaccharides and polysaccharides. In polysaccharides also we have two differentiations based on the monosaccharide that is present, like homopolysaccharide and heteropolysaccharide. Here if you see, high molecular weight carbohydrate polymers made up of monosaccharide units, they are joined by glycosidic bonds. Usually these are tasteless, insoluble and non-crystalline form. Fine, right? These are important. What are different types of polysaccharides? We have two types, one is homopolysaccharide, second one is heteropolysaccharide. Important ma, what are the bonds that are included and what are the examples of homopolysaccharides and heteropolysaccharides. This is important for your MCET and NEET also to remember the examples. One type of monomer, for example, starch, glycogen, cellulose and inulin, which is the polymer of fructose. These comes under what? Homopolysaccharides. Heteropolysaccharides we have two or more types, for example, pectins, hemicelluloses and glycosaminoglycans. Okay, these are the examples of heteropolysaccharides. Next, starch. In plants they act as storage component in plants is starch. Homopolymer of glucose with two components, here it has two components. Amylose is linear helical, this gives what color? Blue color with iodine. Fine, right? Amylopectin is branched, this is main energy reserve in plants. Amylopectin is branched and this is the main energy reserve in plants. Glycogen, animals, fungi, storage. Glycogen is storage carbohydrate in animals, fungi. Highly this is branched glucose polymer and this is main reserve for carbohydrate in liver and muscle cells. Now, inulin you will see in the as storage component in Asteraceae family, wherein fructose homopolymer is stored in tuberous roots and stem, for example Daila Jerusalem artichoke. Next, chitin which is present as the exoskeleton of arthropods, which is a component of exoskeleton in arthropods, fungi and it is also the structural component. Here it is a polymer of N-acetyl glucosamine which forms exoskeleton of arthropods and fungal cell walls. Functions. This provides energy storage, glycogen, starch and inulin, structural support by cellulose and chitin. Cellulose is one of the major component of plant cell wall. Chitin is the main component of fungal cell wall and arthropod cytoskeleton. And matrix or adhesion roles like pectins and hemicellulose are present in the cell wall component of plant cells. So, here if you see, first you explain about polysaccharides, then you explain about the types of polysaccharides. It is okay if you are writing in this way, or it is completely fine if you draw a flow chart also, homopolysaccharides and heteropolysaccharides. Like that also you can describe like in a tabulated form, fine, right, but make sure that you are mentioning all the points most accurately. You are mentioning all the points most accurately in your answer. Now, what you have to do, as I told you, immediately after looking into any set of short answers or long answers and practicing the answers, immediately go for the objective practice. Okay, that is how you will maintain your objective practice and answers on the parallel track. Fine, right. Now, cell cycle and cell division. Here if you see, explain prophase 1 of meiosis. Prophase 1 of meiosis. Asked multiple times like May 2024 and 2023, 2019, 2017 and 2015. These many times the question is asked. Prophase 1 is the longest phase and most eventful phase of meiosis 1. This is the most eventful phase of meiosis 1. Homologous chromosomes will pair, they will exchange the segments and prepare for their separation. It has 5 substages in it, okay, 5 substages. Leptotene, it has thin threads, chromosomes begin condensation and they will appear as long thin threads with bead-like chromomeres. Sister chromatids are not yet distinct. Next, telomers often cluster at the nuclear envelope. Bucket arrangement is seen. This helps in next step pairing. Okay, this is about prophase 1. So, first I will here itself I will explain you few points wherein you can pick them up and present in your answer. Here itself I will be explaining you. Prophase 1 of meiosis is a hot spot topic for your MSET and NEET as well. So, whenever you pick up this stage prophase 1, it has 5 substages, right ma? It has 5 substages starting with leptotene, second zygotene, followed by 3 pachytene, then diplotene, followed by diakinesis. Fine, right? These are 5 substages. First in leptotene if you see, the chromatin material will untangle to chromosomes and here chromosomal condensation will happen. Asters will move towards the opposite poles radiating out the spindle fibers, and in this stage you can see a spireme stage. Spireme stage or bucket stage it is called as chromatid threads will get attached to the nuclear membrane. Fine, right? And then if you see leptotene, these stages are clear right now. Spireme stage, asters will move towards the opposite poles. Chromatin condensation will happen into chromosomes. Fine, right? This and all will happen in the first stage. And this chromatids will be visible under the light microscope. In zygotene stage, the chromatids which have same alleles on them addressed as homologous chromosomes. Pairing of homologous chromosomes will happen and formation of synaptonemal complex occurs in the zygotene. Remember, synaptonemal complex formation which is also addressed as synapses, formation of bivalent, occurrence of tetrads, formation of homologous chromosomes occurs in the stage zygotene. Next is pachytene. Pachytene is the stage where recombination will occur, and these 2 chromatids of 1 homologous chromosomes are addressed as sister chromatids. These 2 chromatids of 2 adjacent homologous chromosomes are addressed as non-sister chromatids. Here in pachytene stage what happens ma? Recombination of non-sister chromatids of homologous chromosomes will happen over here. okay, recombination of non-sister chromatids of homologous chromosomes, which is known as crossing over, and this is an enzyme mediated process. The enzyme is recombinase. Followed by diplotin. Okay, diplotin, this is the stage wherein few cells like mammalian oocytes will stop their cell division. Okay, like mammalian oocytes, they will stop their cell division at diplotin stage, wherein that stage is addressed as dictyotin stage. Dictyotin stage, and here in diplotin, the stage itself will begin with dissolution of synaptonemal complex and formation of chiasmata. What is chiasmata? As synaptonemal complex disintegrate from each other, they will form an X-shaped structures like this. X-shaped structures are formed. These are addressed as chiasmata. Next, last stage is diakinesis. In diakinesis, the stage itself will begin with dissolution of chiasmata, and the ultimate point that you have to mention is disappearance of nucleolus, nuclear membrane, and Golgi bodies will happen in diakinesis, which is the last substage of prophase 1. Prophase 1 is the lengthiest phase in meiosis. Okay. So, these are the points to be remembered. This you can present it in your own terms. Okay. Maintain proper terminology, present your answers in an accurate point wise manner. You will be benefited not just for this IPE short answer, it will be there for your MSET and NEET also. This is the hotspot question, which covers 3 exams. Okay. Next, zygotine paired threads. Synapses, homologous chromosomes pairing. Okay, synapses, homologous chromosomes pairing. This precisely along their length assisted by synaptonemal complex. Each paired set will form bivalent. Here you can mention what? Bivalent, tetrad, homologous chromosomes, formation of synaptonemal complex. Everything happens in zygotine. Next is packetine, as we have discussed. Recombination occurs between non-sister chromatids of homologous chromosomes, which is an enzyme mediated process. Next is diplotin. Here this begins with dissolution of synaptonemal complex, and this will remain at what? Chiasmata. Chiasmata will indicate the place where crossing over occurs here. Next, diakinesis. Chromosomes reach maximum condensation. Chiasmata will terminalize and they will move to the opposite poles. Next, here nucleolus, nuclear envelope will disintegrate. Meiotic spindle assembles, preparing bivalents for their alignment for the metaphase 1 stage. I recommend to draw this diagram at the beginning only, because this stands as a strong reference for you to attempt the question in a systematic pattern. Fine, right? Just look into all the stages. If you are having any doubts, just ask me, ma'am. Now, next question will move on to differentiate between the events of mitosis and meiosis. Very important question actually. Yeah, here we go. First, starting from site of occurrence. If you see, mitosis will occur in the somatic cells. Site of occurrence, mitosis will occur in somatic cells. Meiosis will occur in germ cells for sexual reproduction. Number of divisions, 1 division per nucleus. 2 successive divisions will occur in meiosis, one after the other. Okay. Prophase, simple and short without synapses. This is long and complex prophase. Just now we have seen, right? 5 substages in prophase 1. Next, homologous pairing. Homologous chromosomes, they do not occur in pairs. Here homologous chromosomes undergo synapses, forming what? Bivalents or tetrads. Crossing over is absent in mitosis. Crossing over is in between non-sister chromatids of homologous chromosomes. Next, centromere division. Here centromere will split during anaphase. Centromere here they will split only during anaphase 2. Anaphase 1 of meiosis, what happens? two sister chromatids of homologous chromosomes, they will move towards the opposite poles without any centromere splitting. Then number of daughter cells, this produces 2 daughter cells, here 4 daughter cells are formed at the end of meiosis. Okay. Next, another set of differences, ploidy of the daughter cells will be same as that of parent cell. Here chromosome number gets half, that is why this is also addressed as what ma? Reductional division. Genetic constitution, this is genetically identical to the parent cell, whereas here this is genetically different due to crossing over and independent assortment. Next, spindle apparatus, one spindle apparatus is formed over here, 2 successive spindle apparatus are formed because here 2 times the karyokinesis and cytokinesis are happening, one in meiosis 1 and the second in meiosis 2. Next, last is function for growth, repair and asexual reproduction. For growth, repair and asexual reproduction, here for gamete formation and genetic variation in sexual reproduction. Fine, right? So, these are the clear differences between mitosis and meiosis. Fine, right, ma? Now, though redundantly described as resting phase, interphase does not really involve rest. Okay. This has appeared in 2022, 2020, March 2017 and 2016. Okay. Here we have to highlight all the stages that are present in interphase 1. Taking this diagram itself, I will explain you what all are present. Okay. So, interphase first begins with G1, S and G2. Look at this and then go with the theory. Yeah. G1 phase, which is also known as post mitotic phase or pre-synthetic phase, followed by S phase, which is post G1 phase or pre-G2 phase, followed by G2 phase, which is known as post S phase and pre-M phase. We will look into the events that are occurring. G1 phase is the phase which occurs, okay, for the maximum time. Here cell size will increase, cell size increases, most of organelle duplication will happen, most of cell organelle duplication happens here. Fine, right? And cell is metabolically active. Now, when cell is passing from G1 phase to S phase, there is a checkpoint called as G1S checkpoint, which is a major checkpoint. In S phase or synthetic phase, 2 events will happen, one inside the nucleus. What is the event? DNA or genome duplication, and one in the cytoplasm, centriole duplication. Clear, right? This is followed by G2 phase. In G2 phase, semi-autonomous organelles or self-duplicating organelles like chloroplast and mitochondria will duplicate. Along with these 2 organelles, Golgi bodies also replicate in the G2 phase, followed by tubulin protein synthesis. Fine, right? So, this is G1, S and G2. Here in G1 phase only, if you see, those cells which are not continuously dividing, those cells which rarely divide, such cells, they will exit G1 phase and they will enter into G0 phase. This G0 phase is addressed as quiescent center or quiescent stage. Whenever they want to resume back to the cell division, again from G0 phase they will resume back to division to G1 phase, from G1 to G0 followed to G1 again, fine, right. So, this is what you have to explain in a sequential manner, like what are the events that are occurring in the each phase of cell cycle. If you are having any doubt, just ask me, just go through this chart for 2 minutes, and if you are having doubt, just let me know. Fine, right. Then these are the key features of interface G1, S and G2 phases. Fine, right? Yeah. So, this ends our concept of short answer type of questions from first year, and then after Pongal we are going to start very short answer type of questions by exercising PYQs as well. Okay. VSAQs, most of the students will lose your marks in VSAQs only, very shorts, right, because you will not have at least 1 or 2 questions of the main prioritized questions. Okay. Prioritized questions will not be there in VSAQs. Mostly you will be losing your marks over there, and if you exercise VSAQs strongly with objective practice, that gives you a good confidence for your MCET and NEET as well. Okay. So, post Pongal get ready for learning VSAQs, EAP set and NEET questions parallelly. Okay. So, I wish you good luck, prepare well and get ready for the objective and subjective exams parallelly. Fine, right, ma?